Okay, thank you.
Yeah.
So thank you very much, Nita, for the introduction. Okay, so once again, my name is Hannah, and as what Munta said, as part of what Munta said, we are having the forensic pulse. So the forensic pulse is an organization created to create awareness about what forensic science is all about. So we are having online lecture series from time to time. Okay, so with the online lecture series, our topic number one will be on forensic DNA analysis. And forensic DNA analysis is quite broad. Okay, so we'll narrow it a bit further to paternity testing. Okay, so welcome, and I hope everyone will gain what they seek to gain. Whatever knowledge you want to gain, you would, by the end of the lecture, all of you will be fulfilled. Okay, so with that much I do, let's start. But before we start, let's try to brainstorm a bit. Okay, I know somehow you've heard about DNA in one way or the other. Okay, so I want us to be interactive enough. We should try to engage and in quotes, nothing is a bomb here. Okay, don't say that, oh, whatever I'm going to say will be like a bomb. It's never a bomb. Even if it's a bomb, definitely we are not going to die. It's not a Russia bomb or whatever. So just feel free and let's communicate. Okay, so let's brainstorm. What do you know about DNA? I know one way or the other you've heard about DNA. So what do you know about DNA? Anyone? Can anyone tell me, please? Am I audible?
Yes, yes,
Hello.
Yeah, we can hear you.
yes, you are
We
very
can hear you loud and
audible.
clear.
Okay, so please, who can tell me about DNA? Let's just give a general idea. You hear people talk about DNA, each passing day. As I said, don't be scared. Let's try to contribute and engage with each other. Okay, it's a lecture, it's not presentation only. So let's try to engage with each other. So, what do you know about DNA? If we say DNA, what is DNA at all? Anyone to tell me? Or should I mention names?
Yeah, hello. Hello.
Yes, okay.
Okay.
Hello, yes, I can hear you.
Okay. Basically, I understand DNA to be the genetic makeup of an individual or of a person.
Okay, okay, all right. So thank you very much. Okay, yeah, what you said, it's really makes sense. It's very true. Okay, so as we go on with the lecture, you get to know much about it. And I also like to ask, so thank you. Your name, please.
Benjamin.
Okay, Benjamin, thank you very much. All right, so the next question, which one? Okay, is it blood grouping
Um,
tests? And you have to give your reasons for your answer. So anyone to tell
he-
me
he-
blood grouping, DNA test, which one will you prefer
Hello,
for
Hannah.
to
Hello,
prove
Hannah.
paternity?
Your slide is
Your
static. It's
slide is
not
static.
moving.
It's not moving.
Could you
Could
check
you
that?
check that?
Oh, I've moved.
Yeah.
Okay, I'm coming.
No, instead of yeah, could you display it like full
Played
screen?
like full screen.
Say it again.
Is it visible now?
No.
Munta.
Yeah, it's
Yeah, it's
visible now. We
visible
can
now. We
see
can
it.
see it.
Yeah, it's visible.
Okay, all right, thank you. So the next question I was asking, which one will you use for paternity to prove your paternity? In other words, is it blood grouping or DNA test?
DNA test.
And
But I
why?
prefer DNA test.
Yes. So
So
please
please
tell me
tell me
why.
why.
You
There is
use
a
uh-huh.
total genetic makeup.
Mm-hmm.
We, and it takes 1% of our DNA stores all the information from a paternity and a maternity, or the from maternity it contains in mitochondrial DNA.
Okay. So the line was breaking. You said because what?
Sorry.
Yes, please come again. What did you say? The line was breaking, so I couldn't hear you. Hello, am I audible?
Yes, we can hear you. I think the person who was
Yeah,
asking the
we
question is
can
not
hear you
online.
clear.
Yeah, so if
Okay, so if anyone else can answer that, I'll appreciate. Okay, so in other words,
Yes,
normally we use
so
it's DNA. Okay, okay. Yes, come on, come on.
yeah, so the DNA is that individual specific material that uh, can properly identify humans, plants, animals, and other living organisms, even if they do not want to be identified. So, using the DNA for identification will be the top priority if you are to select.
Okay, thank you very much for your contribution. Okay, it makes sense. So I'll add to what you said. Okay, we have different groups, blood groups, being it AB, O, and other. But for blood group, it's quite general. So about when it comes to DNA test, it's very conclusive because DNA is unique, and before an individual is formed, your mom need DNA. They'll give you your
So with
complete DNA. DNA
so with
is
DNA is
specific. It's
specific.
either
It's
you
either
you are the
you
child
are
of
the
a
child
particular
of a
parent
particular
or not.
parent or
But
not.
with blood
But with
grouping,
blood
so
grouping,
suppose
so
let's
suppose
say
let's say
two
two
alleged
alleged
fathers
fathers
come
come
to
to
blood group, say that the child having blood group A, it means both fathers is quite broader, but it gives an idea of whether the possibility of a child relating to a I know you've heard when we talk of things that are very unique to each individual, our fingerprint is very unique. No one has my fingerprint, no one has your fingerprint. Our fingerprints is very unique. So is to point. Or do you think one is more unique than the other? Any idea?
All right. I think it's fingerprints because
Why?
for
Okay.
identical twins, for identical twins, the probability that their DNA might be identical is very very low. But with fingerprints, it'll be very 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 low. But fingerprints, it's still different among them.
Sure, sure. Yeah, so exactly when it comes to
Hello.
identical twins. Hello.
Can can I answer you?
Yeah, sure, you can. You can also
I
add
think
your. Mhm,
I think DNA is more unique than fingerprints because in some
mhm.
cases or in our research, fingerprint are same in a twins, but DNA, the STR every child. So I think so DNA is the unique as compared to fingerprints. Thank you.
Okay, thank you. Well, I beg to differ a bit. I don't know why you're saying DNA is most. They are all unique in any ways, but when it comes to identical twins, okay, because they are from the same zygote, which divides when it comes to the division process. So they divide, but they are from the same zygote, okay. So when it happens this way, identical twins have the same set of DNA, but when for fraternal twins, they will have different sets of DNA. Okay, and when we say something is unique, meaning is different as like the whole world. You are the only one having that. You get it. So I would say fingerprints is more unique compared to DNA, but DNA helps in various ways because it's much robust as compared to fingerprints, especially when it's latent fingerprints. It can be lost easily, but DNA is there. It's just there. So anytime you can use it. Okay. Anyway, thanks for your contribution. So I think I've even answered the next question, which is in what instances in crime scene investigation would the forensic scientists prefer to use DNA instead of fingerprint? Okay, so fingerprint sometimes can't trace evidence. It can lost easily without any, especially when it's latent. So then DNA is more preferred in this instance, and many other, they all work hand in hand. At a particular time, you prefer to use this over the other, but they all work in hand. But when it comes to fingerprint being latent and getting lost easily, we prefer to use the DNA. Okay, I hope I make sense.
Yes.
Sure. And let's also brainstorm on this. What are the sources of DNA evidence, or where can we get DNA? Anyone, someone tell me, so what then is DNA and where can we get DNA? Anyone to tell me? Please contribute. What are the sources
Sorry, can
of
you repeat?
DNA? Yeah, yeah, sure. Please contribute.
Can you repeat the question, please?
Mm-hmm.
Hello, am I audible?
Yeah, sure, you're audible.
Program, not pick all the yeah, close.
Yes, from
Okay.
blood sample, yes. And what else?
From
And
saliva,
semen
Ah,
sample
okay, okay. Should have been batch.
Saliva, perfect. What else?
and
Hair.
semen fluids.
nail,
Perfect. Yes, nails. Yes. So all what you
Hair.
Uh,
have said, yes, correct.
Madam,
Bone.
Correct.
I'm
And it is
currently
from hair also.
working on a touch DNA. So where we are touch, so we can get the DNA.
Sure,
This is
sure,
not
sure.
necessary for blood.
This is not what? Okay. So thanks for your contribution. They are all correct with the sources of DNA evidence. Okay. So well done. So now with the summary. So what we aim to achieve at the end of this lecture is summarize the DNA, process of DNA, we get it from either the nucleus or the mitochondria. Okay, so we then have to talk about what is nuclear DNA and mitochondria DNA. All right, and we also talk of the reasons for DNA analysis. Then we come to paternity in the procedures in DNA analysis. So we give an overview of the seven main procedures and the SCL loci DNA markers, what it is and how it helps in the DNA analysis. Okay, so these are what are the suggestions you guys gave that you want to know much about. So at a point when it gets too technical, it's a way to help satisfy everybody here. You know, some are here, they want to know more, some too are now beginning to understand what DNA is all about. Okay, so let's try to contribute and help each other. Okay, so forensic DNA analysis. So forensic DNA analysis is an investigative technique. It's an investigative technique because it helps in crime detection. Okay, and this employs the use of DNA profile. DNA profile here is a unique like a unique patterns showing the characteristics of DNA. So my DNA profile is different from, let's say, Benjamin here or whoever is here. Everybody has a different DNA profile because our DNA is what is unique. What DNA profile that is? It's established the links between biological evidence and tries to compare the samples. Okay, so looking at the right side of the slide, so supposing you get a biological sample from a crime scene, okay, and you perform DNA analysis to get a DNA profile. So this is a DNA profile, simplified form of DNA profile, and you try to compare their profile to some suspects you think they could have any link with the crime scene. Okay, so you can't just link someone without evidence. You know, when it comes to the court of law, they want evidence. Evidence does everything. So let's say we have three suspects here, okay, and looking at the profile, you get the profile from the crime scene and the profile of that of the suspects involved. So here we have three suspects. So who can tell me with the crime scene sample serving as the reference sample? Who can tell me either suspect one, two,
Hannah,
or
please, your slides are not
hello
moving.
Please, your slides are not moving.
again?
Can you use the presenter
Can you
mode
use the
instead
We
of
can't
presenter mode
see the
instead of
slides.
the?
yeah?
Yeah.
So can you see it now?
Yeah, and can you click to go to
Yes,
the
and can you click to
slide
go
Okay, sure.
to the slide view, slideshow view
yeah,
to expand the
All right.
distance?
I wanted to expand it, but if this is visible, then that's fine. Okay, so it's visible now,
No,
right?
put it in slideshow. Put it in slideshow so that it could be bigger.
Slideshow. Which one is that? Let me see. But can you see it now?
Yes.
Yeah, but
Yeah.
the get
Okay. Okay. All right. Sure.
I
Sure.
it's manageable.
So
So
okay, fine. So I guess I have to explain again. So let's say you get a sample from a crime scene. Okay, and you do DNA analysis to get the profile of it. So this is a simplified form of a DNA profile, and you are suspecting, let's say, three people to have some connection with the crime scene. Okay, so you also do DNA analysis to get the DNA profile of these three suspects. So these are also profile here. So who can tell me whether crime scene sample serving as a reference sample is it suspect one, suspect two, or suspect three that has something to do with the crime scene? Yes, anyone?
One.
Suspect yes, very correct. Because as you can see, the DNA profile matches exactly like that of the crime scene sample. So with this, the outcome could be used in the court of law. Okay, so as a forensic scientist, it's very important and very vital to work, you know, meticulously so that your whatever result you will get it's accurate because you can't end up complicating someone's life because of your mistake. Okay, So this what forensic DNA analysis generally is all about. So continuing, is my slide visible? Sources of DNA evidence.
It's not moving. It's not moving. Can you?
Not moving.
Yeah, I think you should be moving the slide on the PPT.
Okay, is it visible now?
No, no,
No,
no.
no, no.
Okay,
It's still on
I
voice.
don't know
Yes.
why it's
Oh yeah,
Yes,
now,
yes,
now,
yes,
now
it's
you
moving.
can see it now. Yeah.
wow. Then I have to do for each one like that. Okay. So sources of DNA evidence. Is it visible now?
Yes, yes.
It's full now. It's
Yes,
full
it's
now.
moving.
Thank you very
Thank
much.
you.
So sources of DNA evidence, or we get DNA from biological samples, and you've helped me enough. You've mentioned some of the DNA where we get DNA. Okay, so you can get it from the tooth, the bone, the hair follicle, and for the hair follicle, okay. When you pull up the hair, when the root is attached to the hair, you can use it for what we call the nuclear DNA analysis. And with the shaft of the hair, we can use it for mitochondrial DNA analysis. We'll talk about the differences later, like what's nuclear DNA and what's mitochondrial DNA. Okay, so just stay tuned. And we can also even get it from urine, and we can get it from semen, then saliva and blood. Okay, so looking at the right hand side of the slides, we have types of sample where we can get DNA, and the amount of DNA we can get from these samples.
From the stool also.
Audio is lost.
Hello, guys. Hello. Yeah, I think she's having some difficulties in the network. So as we wait for her, let's just do a little recap of what we have learned so far. Okay. So as she joins, so from the beginning, so from the head to toe. So what have we learned so far? Anyone?
Okay. Um, what I've learned is that she was saying that
Sorry.
with the base, we have this base of DNA.
I've also learned that fingerprints are more unique than the DNA.
Okay, fingerprints are more unique than DNA. I think I remember she did mention in the case of um, identical twins.
Yes.
So in some cases they are both unique, both fingerprints and finger DNAs are both unique. But in the case of differentiating between identical twins and fraternal twins, that is when the aspect of fingerprints will come in.
Yes. Yeah. Hello.
Yeah, hello. So Benjamin.
Yeah, yes.
Okay, I know fingerprints has its strengths and weaknesses. DNA has its strengths and weaknesses. So considering both of them as forensic evidences, like which of them do you think how like call it has the highest
Yeah.
distinguishing.
Hello.
Yeah, hello. Yeah, Mark, sir.
Yes. Munta. Good
Yeah,
afternoon.
yeah. Good afternoon.
I was following until Hannah. I was following until Hannah went off.
Yeah, yeah, yeah.
So yeah, when she was asking the questions, I wanted to add my suggestions regarding the DNA and fingerprint.
Okay.
Uh, in terms of uniqueness, no one is superior over the other. Okay, DNA and fingerprint. And then in terms of identification, also depending on the context in which you are looking at it. If you are looking at it for, uh, I mean, crime purposes, solving crime and then attaining justice, also, then you can't choose one over the other. Should you find both of them at the crime scene? Okay. The limitation is that, as it stands now, fingerprint is subjective as compared to DNA. So, therefore, if you obtain fingerprint at the scene of crime and you perform all your examination, the decision as to whether it is uh, originating from this person or not lies solely with the examiner's view, okay, cognitive purposes, as compared to the DNA, where the technology would give us the comparative result without any need of the examiner's subjectiveness. All right. So that is the only time you would want to choose DNA over fingerprint, and also the rare cases wherein the identical trace comes in. All right. So then again, it depends on the context and how you want to use it. But for uniqueness, both of them are very unique. It depends on how you would collect them, preserve them, and then examine them. Okay.
Sorry,
So
can you
that
repeat
is.
that? Thank you. Please, can you repeat those unique distinguishing uh,
I'm I'm I'm
factors?
saying I'm saying that for education purposes, now it is even wrong to say that fingerprint is unique. There are actually unique features in the fingerprint that makes it unique. So if you want to describe fingerprint in terms of uniqueness, then you would have to specifically say that maybe the rate characteristics or the minutiae in the fingerprint varies from person to person, rather than saying that the fingerprint are unique, because fingerprint, as you would mention it, would mean a lot of things. Okay, would mean a lot of things, because we have the fingerprint pattern type, that is the loops, hole, arches, and the other types. Those ones are never unique, which is why the distribution of these fingerprint pattern types remains the same, has been the same throughout, since its discovery. So we know that the loop pattern is most prevalent, followed by the hole pattern and the arch pattern. Since the discovery of fingerprint, this prevalence has been the same throughout, which means that the pattern types are not unique. They keep on passing it from parent to offspring and all that. So it is the unique features in the fingerprint pattern types that varies from person to person. So those are not genetically determined, right? Now, with that of the DNA, DNA is also unique, just like fingerprint. So in terms of superiority, no one is above the other. But then if you want to weigh their odds in terms of weaknesses and strength, then you would definitely go for DNA. DNA, because DNA, the final decision on the DNA examination is not subjective. You know, you run the profiles, and then the outcome of the profiles clearly tells you whom the sample originated from. If you could see from the sample that she showed, most of us are not biology persons, but just by looking at the bands, we're able to tell that it's suspect 2 or suspect 1, right? But then for fingerprint, regardless of how unique it is, at the end of the day, it is the one doing the examination that is going to decide or tell whether, yeah, it is actually unique. Because how many ranges are you finding to be able to say that, okay, it is enough or it is sufficient for you to say that it is originating from this particular person or that particular person? Okay, so if we want to weigh the positives and the negatives in terms of advancement and technology, then of course we will go for DNA. But then, as crime scene aspect, okay, when we go to the crime scene, finding these two evidence at the crime scene is an advantage to us. So we would always collect the two. We wouldn't collect DNA because we think it is better than fingerprint, and then we wouldn't collect fingerprint for the same purpose. We would actually collect the two. Okay. So again, the last thing I would like to say is that, you know, we have to understand this whole connection between evidence, suspect, and then the identification. Finding any of these evidence at the crime scene, I keep saying this everywhere I go. When you find the evidence at the crime scene and you analyze and you realize that it is originating from Mr. A or Mr. B, at that instance, it does not indicate that Mr. A or Mr. B committed the said crime. It only indicates that the Mr. A or Mr. B was present at the crime scene. It ends there. Okay. So it is the type of crime scene that would bring further questions as to what Mr. A or Mr. B was doing there. Because Munta, for instance, is my friend, and I go to his house every day. So if you go to Munta's house and there's a crime and you find my DNA or my fingerprint there, it does not mean I was involved in a crime. He's my friend, so I go there every day. So then the possibility of me leaving any of these evidences at the scene of crimes, according to Lucas principle, is very high. Okay. So once we find these evidences, and we examine and we notice that it is coming from A or B, as forensic scientists, we have to be objective. So at the first instance, it only means that Mr. A or Mr. B was present at the crime scene. We leave it there. Okay. It is originating from A, indicating that he was at the crime scene, but as to whether he committed the crime or not, we would need far more evidences, both eyewitness, ear witness, and other evidences. Now there's CCTV also to prove the involvement of someone in a crime. Okay, because one thing I learned was that it is always better to let an innocent person, a criminal, go than to rather convict an innocent person, right? So that is just what I would like to add.
Okay, okay. Thank you very much for the contribution.
Thank you very much.
No, that was very, very educative. Thank you.
Yeah, a lot of information in that.
Is it visible?
Yeah,
It's
it's been
visible.
Okay, thank you. So we're talking of sources of DNA evidence, right?
Yeah.
So these are the types of sample. When you look at the right hand side, I think that's where I got to and started. So the type of sample, our biological sample, where we get DNA, and the amount of DNA we can get from such sample. Okay, so I was asking a question and it went off. So let me ask again. So looking at the amount of DNA we can get from these samples, okay, which one do you think we can get high amount of DNA? So I want you to just have a quick glance and tell me which of the
Liquid
sample.
semen. From the semen.
Yes,
Liquid semen.
yes, perfect. So perfect.
Followed by liquid blood.
Okay, all right. Thank you very much. Your name, please.
I am Timothy. I'm from
Okay,
Nigeria.
thank you so much, Timothy. Oh, I love Nigeria. Thank you very much.
Okay, you married us. You married
Yes,
already.
yes, yeah. Okay, I love Benna Boy. That's by the way. So looking at this liquid semen, okay, so you can see it has high amounts of DNA, as compared to the rest. But even with the rest with low amount of DNA, we can do what we call the So I think we got to this part. Is it visible?
Yes.
Okay, thank you very much. So what is DNA? Okay. So someone asked what at all is DNA? Where can we find DNA and all that? So DNA is an abbreviated form of what we call the deoxyribonucleic acid, and it's a hereditary material, meaning it's passed down from parents to children. Okay, and in other words, we inherit them from our parents, and nearly every cell in a person body has the same DNA. So let's say when you compare your cardiac cell, it has the same DNA content as your muscle cell and any other cell in your body. Okay, but what brings about the variation is with the function and structure of that particular tissue or cell we are talking about here. So we have what we call the gene regulation and expression of gene to bring about what the function is supposed to do. But every cell in the body has the same DNA content. So if I pick, let's say, your liver cell, or I pick a sample from your liver, it should have the same DNA as when I take it from other parts of your body as well. So this is what it basically means. So where is DNA located? So DNA is located in human, okay. DNA is located in the nucleus and what we call the mitochondria. Okay, so mitochondria. So we have come to talk about the nucleus and then we talk about the mitochondria later. Am I audible?
Yes, you are. Yes, you are.
Okay,
Yes,
all
you
right.
are.
Perfectly. Very clear.
Okay,
Yeah,
thank
very
you.
clear.
So the nuclear DNA is about 3.3 billion DNA base pair. So not to exaggerate, let me just give an analogy. Okay, so it's like a thread. When you pull a thread, okay, it can move from down to the sky and down because 3.3 billion is very long. So one will ask, in such situation, how is it possible for such a long thing to fit into nucleus, this nucleus, okay, in the cell? How is it possible for DNA to fit into the nucleus? Okay, but nature has it that, or it has it that the DNA, okay, so this DNA, when it's in the thread-like form or when it's elongated, the DNA winds around a protein called the histone to make it compact, okay, into what we know as the chromosome. So we have 23 pairs of chromosome, in humans. So with this, it can fit perfectly into the nucleus. All right. So also when you look at the basic level of DNA, what it contains, the basic unit of DNA is we have what we call the nucleotide. So the DNA, okay, it looks like a ladder with the horizontal steps having what we call the nitrogenous bases. Okay, so these are the nitrogenous bases, and mostly the guanine, okay, combines or bonds with the cytosine with three hydrogen bonds, as you can see here. So one, two, three, and the adenine binds with the thymine with two hydrogen bonds, as you can see here. So these are the nitrogenous bases in the middle here. Okay, and when let's say you are comparing it to a ladder, so the size of the ladder, they are made up of what we call the phosphate group. So this round thing here is representing phosphate group, and this is the ribose sugar. So this basically what DNA, the content of DNA is all about. So with this, okay, with this ladder structure, like it then twists, okay, it winds to form the double helix structure, and it winds around these stones to make it more compact, okay, to fit so that it forms chromosome to fit into this nucleus. I hope it's understandable.
Yes.
Sure. Okay, so any question? So this nuclear DNA.
So,
Hello.
yes, I raised my hand.
Hello. Okay, sure. So please, you can ask your question.
Now, this 3.3 billion DNA base pairs, is it
Sure.
that is found in each of the cell or the whole human
Yes,
body?
yes, in each of the cell. Okay, so
Thank
it's
you.
not the whole human body because as you can see here, this DNA is what will call into what we call the chromosome. So even this is for one chromosome, you can imagine. So the 3.3 billion here is for a chromosome. Okay, so they with the 23 base pair, it will be like, let's say, 33.3 billion times the number of it. So it's quite long. Okay, you know humans, we are complex. We are not as simple, we are complex. So with the protein it produces, which affect our phenotypes, the way we look, the way we behave, and all, it all comes from our what, our genetic makeup. So you should know how it should be elongated. It makes sense if it's elongated. Okay, but the point I'm trying to make here is that it tries to be compact so that it fits into the nucleus. So is it understandable now?
Yes.
Okay, all right. Any other question?
Please, I have a question, man. Good
Okay,
afternoon.
sure. Ask. Good afternoon.
My name is Barry. Okay, I saw somewhere with respect to the question Mr. Timothy asked
All
earlier.
right.
I saw somewhere that the human genome has about 3.1 billion base pairs,
Mm-hmm.
2.9% of which encode for protein.
Sure.
Now, yes. Now, my question is now that is it that this statement is saying it with respect to what you said, or that the whole of the human genome is what has 3.1 million base pairs?
Yeah, yeah.
I will need clarity.
Yes.
Is it the
My
whole of is it like the whole of human genome, like everything, or just one cell that has everything?
yes, just for a cell. Just for a cell.
Okay.
Yes.
All right, thank you.
Okay. Any other question?
My hand is up.
All right. Please ask.
Okay, my name is Ransford.
All right.
So earlier you were saying you did mention that our fingerprints are very unique than the DNA. So I want to ask, why then do parents go on for this DNA test rather than the fingerprints? Because if you did make a statement or a scenario that identical twin use that statement, so I just want to know, why should parents go in for a DNA while the fingerprint is more unique than that?
Okay, thank you, Ransford. So me saying that the DNA fingerprint is more unique in the sense that DNA is also very unique. Okay, um, but comparing our fingerprints is different from each individual. But when it comes to DNA, among identical twins, they are the same because they are from the same zygote. Okay.
Okay.
Now, what you were asking is why is that we don't use the fingerprint, but we use DNA? We can't
Mm-hmm.
use fingerprint because we don't inherit fingerprint from our parents. Okay,
Okay.
fingerprints it's formed from the environmental conditions in when you are in the womb, the environmental condition and other factors. Genetic will form a factor, but it's not main. Okay, so with the environmental condition and due to development and all that, your fingerprint becomes different even from your mother. There's no way you could inherit completely fingerprints from your parents.
Okay.
Okay. Yeah. What I can say, maybe the type of fingerprint. You have types of fingerprints. The loop, the hole, the arc finger. I won't go into details. Okay. Maybe you can inherit the type of fingerprint, but the exact ridges or the exact ridges of your fingerprints can never be inherited from your parents. You get it? So
Yeah.
in
Yeah.
other words, DNA is still the best when it comes to looking out for relationship testing as compared to fingerprints. So fingerprints is more useful when it comes to forensic analysis or crime scene. You want to make it maybe unique. You can use DNA rather, but when it comes to identical twins, that's the emphasis I want to lay here. When it comes to identical twins have the same set of DNA. You get what I'm trying to say? Yeah.
Yes.
Hope it's
Yes.
understandable.
Yes, please.
Okay. Thank you very much. So,
A second one,
okay.
follow-up question.
Yeah, sure.
Yeah,
Ask.
when I did, you did mention that source of DNA test, it can be like blood, hair, saliva, nails, urine. I just want to ask, so
can you use two also for the DNA test?
Yes, perfectly. You can use two. Okay. It's
Wow.
not there, but it is. You can use two.
Okay.
So
Okay.
let's say you go to a crime scene and the only thing available is two of feathers. You should use it. Okay.
Okay.
you
Okay.
you can use it. Yes. Yeah. Sure.
Okay. Thank you very much.
You're most welcome. So can we proceed? Any other question? Okay. So to continue, um, is this slide visible?
No.
Okay. All right.
Yes.
Okay,
Yes.
sure. So the other location where we can find DNA, is it visible now?
Yes.
Yes.
Sure. So the other one is what we call the mitochondria. We have a lot of mitochondria in a cell. Okay.
Mm-hmm.
So the mitochondrial DNA here is made up of 16,569 DNA base pairs. So compared to the nuclear DNA, it has a lot of DNA base pairs as compared to the mitochondrial DNA. But one thing we have to note here is that for the mitochondrial DNA, it is maternally inherited. So none of us inherited our mitochondria from our dads. We all inherited our mitochondrial DNA from our mother. Okay. And who can tell me the reason why? Why do you think we inherit the mitochondrial DNA from our mother and not our dad? Anyone?
I think basically because they carry us in their womb for 9 months. I mean, that's what I can say.
Okay, okay, all right. Thanks, Ransford. It's yeah, it's a contribution. I appreciate that, but there's more to it. So who can tell me?
Can I try?
Sure, you can.
Yes, I think during fertilization, actually, the tail of the sperm does not really fuse into the egg, and it is the tail that is made up of the mitochondrion. So, which means the mitochondrion, which is present in in the sperm cell, which is actually from the tail, does not form part of the fertilized egg.
Sure,
So
sure.
that is exactly how come the father's mitochondrion doesn't form part of the fetus or the baby's own.
Okay, okay. Your name, please.
Frank.
Okay, thank you, Frank. Yes, so that's right. But I want to make the tail doesn't contain the mitochondria exactly. We have what we call the middle piece. Okay, before the tail is attached. So maybe it can also you can also say the tail do, but we have middle piece before tail is attached. So the middle piece of the sperm is what contains the mitochondria before we have the head containing the nucleus. Okay, so thank you so much, Frank. What Frank said, that's exactly the point. So when there is fertilization, the mitochondria of the sperm doesn't fuse along. So it's only the nucleus of the sperm that fuses with the egg. Okay, so we that shows that we inherit our mitochondrial DNA from our mom. Okay, I hope it's understood.
Yes.
Sure.
Yes.
So, a mitochondrial DNA here is closed and circular. So, in other words, I'm trying to bring out the differences between how the nuclear DNA and the mitochondrial DNA looks like. So, with mitochondrial DNA, it's circular and it's shorter than nuclear DNA. So, from the previous slide, okay, you could see the nuclear DNA was linear. Okay, but when you come to the mitochondrial DNA, it is circular. So from here, you see from the nuclear DNA, it's linear, okay, as compared to the mitochondrial DNA, that is what's circular. So moving on to the next slide. Okay, any question with mitochondrial DNA before we proceed?
No, only that generally the DNA is described according to the pattern of the nuclear DNA. They don't usually mention circular. This detail is very nice. I'll just Maybe because that one is more abundant, that when generally when they are describing DNA structure, they don't mention the circular pattern of the. Maybe that
Okay.
maybe that is the reason.
Hello. I think someone's audio is on. Can you kindly mute it? Okay. So thank you. Let's proceed. So reasons for DNA analysis. Okay. Why would one choose to go through DNA analysis? One is to identify potential suspects whose DNA may match evidence left at crime scene. Okay, so um, you are suspecting someone to have anything to do with the crime scene. DNA can help to identify those potential suspects. Like when we started the slide I showed here, so with like example like this, okay, so it's showing that someone may have a hand. So after that, then further investigation could be done to either acquaint or convict the person. Okay, so that's it. Uh, and another point is it's also used to exonerate person who are wrongfully accused of crimes. Okay, so example is here. This man who is so furious here in the year 1978, Craig Colley was convicted for double murder. Okay, so and in the year 2017, they had this DNA analysis. You know, previously in those years, DNA analysis wasn't that advanced, so they didn't know much about it. So in the year 2017, it was done, and it was found out that Craig was innocent. So you can even the picture difference can show you. Here he's so furious, he's angry. Okay, literally, he's very angry. You can imagine not doing something and they are saying you did it. So you can imagine. But here he was, freed in the year 2017. So one reason for DNA analysis is that. And thirdly, it can also identify crime and catastrophe victims. So it can also link criminals to a crime scene in other ways. And moreover, supposing there is any sudden disaster or anything that, let's say, relatives are looking for their loved ones or dead ones in this sense, okay, it can be used to show any relationship with their DNA profile, one way or the other. Okay. And moreover, it can also be used to establish paternity and other family relationships. So apart from paternity tests, as most people know, most people are more used to the paternity testing, but we have what we call maternity testing. So maternity testing here is when you test for the relationship a mother has with a child. So mother to child, maternity, and we also have siblingship. Okay, so this is among siblings to check whether siblings are indeed related, and also grandparents, child to grandparent, and we have what we call the avuncular testing. Okay, so the avuncular testing is uncle, auntie, okay, to niece, nephew relationship. I hope you get it.
Yeah,
Hello. Yeah, so
yeah,
that's for avuncular. So these are some of the reasons for DNA analysis. I hope it's clear.
yeah.
Okay, thank you. Now let's move ahead. Now we are come to talk about the DNA paternity testing. All right. So what then is paternity testing? Paternity testing is the use of DNA profile to determine whether an individual is indeed the biological father of another individual. So as I said earlier on, for an individual to be formed, okay, you take half the DNA from your father and half the DNA from your mom to form your complete set of DNA. So for DNA paternity testing to be done, when we check the profile of the DNA profile of your father and that of your mother, we will find the DNA profile of your father to the DNA profile of a child. You will find out that the half the DNA of the child matches with half the DNA of the father, exactly half. If there is any relation, it will be exactly. There won't be any omission or anything of that sort. Okay, so that's it. And also, DNA paternity tests are extremely accurate. Someone may ask, is it really accurate? Yes, it's very accurate. So a test can show either 99.9% or 0% probability of paternity. So because of statistics, someone may ask, why are we not using 100%? Because of statistics, normally we compare a particular father to the general population. So due to statistics, we don't want to say it's exactly like 100%. Okay, but if you have 99.9% probability of paternity, it means that indeed you are the father literally. Okay, and 0% means you are not. You don't have any relationship between you two. And also, except in rare conditions known as chimerism. So chimerism is a condition here whereby an individual, okay, we all have one set of DNA, but in chimerism condition, it happens that an individual will have two or more sets of DNA. So you see an individual, but the individual's DNA looks like it's two people's DNA, or let's say three or more. Okay, so when it happens this way, the DNA can show a false positive, but this condition, I repeat, is very rare. And even with this one, there's a way to really prove. Okay, so sometimes when the person is coming for paternity testing, we ask some questions to show that the probability of the person being a chimeric is so low. Okay, so we ask, have you had any stem cell transplantation or anything? When it happens that way, you take the DNA of the donor in stem cell transplant. You take some part of the DNA of the person until some time before the person's DNA vanishes from your system. Okay, so I think as time goes on with other lectures, we'll talk more about the chimerism for you to understand better. Okay, I don't want to draw much time into that, but we'll talk more about that. And also, I want your say on this. Most men are really feeling bad for the way most women are very unfaithful to them and here. So there's one saying, one writing on I think Twitter, saying that DNA tests should be mandatory at birth. Too many of you, meaning we the ladies, I'm inclusive, okay, trying to say that too many of we the ladies are lying about who the father is and messing up people's life, including the child. And they are saying especially in Nigeria. So, Mr. Timothy, yeah, they mentioned your country. They said
Yes,
especially
because
Nigeria.
we are the largest. We are so many. That's
Sure,
why.
sure. Yeah, exactly. So, and also they are saying Jamaica. When you look down here, they said Jamaica has the highest rate of paternity fraud. When we say paternity fraud, it's when a lady, okay, maybe does some analysis and see that, oh, this man is very responsible, and I don't know, either she knows the child, the father of the child, or she doesn't. Okay, but is looking for a responsible man to take care of the child. So we'll be like, oh, maybe this man is the one responsible. Where in actual sense, it's not the man. Okay, so that's paternity fraud here. And from here, it's saying that Jamaica has the highest rate of paternity fraud, followed by Nigeria, then Canada, and then UK and France. So Ghana, I think Ghanaians too, we are there, but others are higher than us when it comes to this instance. So what do you think? Do you think DNA test should be mandatory at birth? The moment the child is, um, giving birth to, we should have DNA tests. Yes, anyone?
Yes, of course.
Yes.
Yes, it should, right?
Yes.
Yeah.
For me, I think it should be mandatory provided the government is ready to absorb the cost.
Yeah, sure, sure. Yeah, and I think gradually, even here where I work now, okay, for alleged father and child, Gene Lab Molecular Diagnostic Limited, we charge just 2,000 Ghana cedi. We have the machines here, everything is available here. Previously in Ghana, there was nothing like that. So they used to, ship it abroad for the test to be done. So we have it here. Okay, we have all them and we do it here. So it's quite cheaper, just 2,000 for alleged father and child. So I think as time goes on, it will even be less cheaper. Okay. So I think it will get to a time DNA might be mandatory at birth to prevent, people from thinking too much, our mental health is very important. And also, not only for the men in this sense, sometimes some men also refuse responsibility for their child. You get it? So in this case too, it can either prove that one fathered a child. Okay, so that women too, you'll be free like singling, handling a child, single mother here and there. You get it? Yes. So any question? Hello, hello.
Hello.
We are hearing, we are hearing you.
Okay, so no question, so I can proceed,
Yeah,
right?
yeah.
Yes.
Yes. All right. So reasons for paternity test. Why do you think someone would do paternity test? For peace of mind. Okay. So one of the reasons is for peace of mind. Supposing you are with your woman and all of a sudden the woman goes like, maybe you are carrier and the woman goes like, do you think even the child is yours? And maybe you've taken care of this child for let's say 15 years, 10 years, or even more. And the woman is saying this. At that instant, trust me, I'm not a man, but I can feel in the shoes of such person. You will be confused, okay, you lost appetite, all those things. So to make your, have your peace of mind, you just have your paternity test. And also to gain legal rights, so let's say custody of the child, inheritance situation, and also maybe a father is refusing to take responsibility of the child, and you want to file for these those things and all that, you can also do it. Okay, so paternity test also counts in this sense, and also identify links to genetic conditions. So we inherit our DNA from our parents. So basically, if you find out that maybe this is my dad, okay, father to child relationship, the probability that if the father has a genetic condition, it will be transferred to the child is high. So in this case, it helps in this situation as well, so that further, probing can be done to solve that situation. And also for immigration purposes, in this case, let's say a father is abroad and wants the child to join. Most country, it's a requirement that you have a DNA test to prove that you have a relation before you can bring your ward to where you are. Okay, so that's the reasons for paternity testing. So now, yes, is the slide visible?
Yes, yes, it's visible.
Okay,
Yes,
so
yes,
now,
it
yeah,
is visible.
yes. So now I would like you to have much concentration here. So what we were saying previously is more like a little knowledge about DNA, but this is more technical. And I added this because that's the requirements or some of the suggestions some of you put across. You want to know more about what goes into DNA analysis and all that. So you
Oh, network.
will talk about the steps. Is what we call the sample collection. If your sample is collected, there's certain things that as a DNA analyst or the person taking the sample, you have to note. You have to try as much as possible to avoid any form of contamination. and it can contaminate your sample. Okay, so you try as much as possible to avoid any form of
Yes.
contamination. All right. Smear or the cheek swabbing. So you get DNA from the cheek cells. So it's a cell. So cheek swab and also blood draw. Can get it from blood draw and saliva sample. And supposing a newborn heel prick and you want to use blood sample, so you can use that for newborns. And we have what we call the amniocentesis. Okay, so this is when a pregnant woman wants to find the paternity status of the fetus. Okay, we use what we call the amniocentesis. So what is done here is uh, with the aid of an embryologist, the amniotic fluid is picked from the womb, okay, and it's compared to the father to see whether they have any relation. Okay, and this is quite invasive, meaning with the slightest mistake, you can have miscarriage. So it's quite invasive. By now, there is a more advanced form that we use blood from the pregnant woman. Okay, to so with this, if a pregnant man wants to use blood, it's very expensive. Okay, because this has a whole lot of procedure with pregnant women. So it used what we call the cell-free DNA. Okay, so with that, we believe with pregnant women, when they are pregnant, at times in their blood, the DNA of the fetus, sometimes, or the cells of the fetus run in their blood. So with that, we can use that and isolate the DNA of the fetus from the pregnant woman's blood so that we get the DNA of the fetus and compare to that of the father. Okay, and this also for pregnant women. There's another procedure known as the chorionic villus sampling, and with this, a snippet of the of tissue is picked from the placenta. Okay, so with the placenta, you get the DNA of the fetus to compare with that of the father, and we have the fine needle aspiration. So with these two, it's normally used in cytogenetics procedure or in situation whereby you want to find for chimerism situation. Okay, you want to compare the cell you picked from the bone marrow, whether it corresponds to other DNA from other parts of the body. Okay, so that's for sample collection. Any question on this? Hello? Hello?
Thank you, sir.
No, please.
Okay, so let's continue. All right. So the
I think
step.
if anyone wants to ask a question, raise a hand or just share it on the chat box. So we'll draw your attention.
Okay. All right. So with the step number 2, we have the extraction. So after you've collected your sample, we do what we call the extraction. And the principle behind extraction is that we want to isolate the DNA from other cellular components. Okay, so and we have different forms of extraction protocols. Okay, so the principle is like this. From this side, we have what we call the tissue dissociation. So the tissue dissociation will sort of disintegrate the cells. Okay, so after you get the individual cells, we call the cell lysis to break down the cell to release the DNA. To get into the DNA, it has to break the cell, then the nuclear membrane, if or the mitochondrial membrane here to get the DNA released. So after you get the DNA released, you have to find ways to isolate the DNA from other cellular components. Okay, so here there's DNA binding. So the DNA can bind to any particle, maybe like silica particles, binds to them so that here it is washed and the DNA binding to the silica particles, the other cellular components are being washed away, leaving only the DNA here. So here with the last step, elution, with your DNA in the elution, so that you can use it for further procedure. Okay, so we have various formats for extraction protocols. We have the Chiagen extraction, we have the chillers extraction, organic extraction, FTA or solid phase extraction. We have differential extraction and direct extraction with extraction buffer. So all this they are based on the same principle. But for example, with the differential extraction, it's normally used in sexual assault cases. Okay, so what it happens is at the end of it with the DNA you get here, it will separate the epithelial cells from the sperm cells. Okay, so that you can further look at them to compare with the suspect to find who is the actual perpetrator of the crime. Okay, so this for the extraction. All right. So any question? Okay. So with the quantification, okay, so after you extract your DNA, hello, hello.
Yeah, I'm with you.
Okay, okay, all right. So with the quantification, after you get your DNA, now you have your DNA, you have to make sure that the DNA you've gotten is enough for you to proceed with your DNA analysis. Okay, so there are various ways you can do that. One is the UV absorbance and uh, fluorescence dyes, agarose gel electrophoresis, capillary electrophoresis, or the nano drop. Okay, so these are the methods you can use to quantify your DNA, and this will prevent wastage or what we call the dissipation of DNA samples. Okay, so let's say supposing you get DNA from crime scene. Normally, the DNA you get maybe in small, form. So you don't want to finish everything, and you find out that the DNA profile you have, the peaks are not showing well, or whatever you want to achieve, it's not showing well, and you find out that you've wasted the little sample you had. So this is to find out that you have enough sample to proceed with other, procedures. I hope you get it. Yes. So that's for the quantification. And then the fourth step is the amplification step. So after you find out that you have enough DNA, you do what we call the amplification. And with this we use the reaction known as the PCR, which stand for polymerase chain reaction. So this will amplify the DNA, or in other words, make various copies of the DNA. Okay, so um, with the PCR components, we have the DNA sample. Okay, so this DNA sample is the DNA you extracted, all right, or in other words, the DNA template for the copying of it. And primers are short nucleic acid that binds to the DNA, so that it serves as a starting point for the formation of new strand. And definitely, if you are trying to make copies of the DNA, you need nucleotides to form these new strands of DNA. And we have an enzyme known as the Taq polymerase. Okay, so this is what helps in the elongation of the new strand you are making. And we have the mix buffer. Okay, buffer here is to make the pH and give out a stable pH, and also give out optimal reaction activity. And all this, you use the PCR tube to do all this. Okay, and the machine, okay, we use for PCR is called thermal cycler. So with the PCR process, we have the denaturing. So with the denaturing is to break the hydrogen bonds. Okay, the hydrogen bonds between this DNA. As I said earlier on, we have made the guanine connecting with the cytosine with three hydrogen bonds, adenine connecting with the thymine with two hydrogen bonds. So all these bonds has to be breaking down. So what is that? We apply heat of 95 degrees Celsius to separate the strands of this DNA. Okay, so after they are separated, okay, the primer attached with the complementary part, and it's called the annealing to serve as the starting point for the new strands to be formed. So with the new strands to be formed, the Taq polymerase binds to the primer, and it's what the nucleotides binds one after the other to form the new strand in this extension procedure. And that one is done at a temperature of 72 degrees Celsius, where the primer binding is at what 55 degrees Celsius. Okay, am I clear?
Yes, please.
Hello.
Yes.
Okay,
Yeah,
so
very true.
all right. So with that, after you amplify, you go through a procedure known as the separation. So with the separation, okay, we use these machines. We can use with the separation, can use agarose gel electrophoresis, but that one I would say it's archaic. It's quite. But the more advanced form, we use the genetic analyzers. Okay, so this is amplified biosystem 3500 watt genetic analyzer, and this is Promega CE system, compact spectrum system. So this is what we use at GenLab, the Promega CE system, right? So on genetic analyzer, the main function of it is to perform sequencing and what we call the fragment analysis. Sequencing here is to know the nucleotides involved, but fragment analysis, which normally we use in the paternity testing, is just to know the sizing, the quality of it, and for other information. Okay, so this is the machine normally that we use. There are more other brands, okay, but the Promega here has higher to a loci marker, so it gives a higher discrimination. That's why we use it here to give accurate results as much as possible. All right. So the next step is what with this machine, it will give you a data. Okay. So after you get the data, we have the analysis and interpretation. It gives out the result for you to analyze and interpret your results. So here, this how when after you are done, this how a DNA profile looks like. Okay. So if someone is new, the person won't understand. But in other words, this is how the DNA profile looks like. So these gray portions on top, okay, with some D8 and co, these are called the DNA markers. Okay, so they are the DNA markers, and each peak um, below it. Okay, so the DNA marker D8S1179. So from this point to this point, any peak under it is for that marker. Okay, and for every marker, we have to get two alleles. Okay, or two alleles. So with that, because we have to get two because, as I said, for your DNA makeup, you take half from your mother and half from your dad. Okay, but in the sense whereby it's homozygous, the allele size is the same. It shows only one peak. Okay, so in this sense, it means that it is 13-13. So for this individual, it is what 13-13. Okay, and here too is what 30-30. And as you can see here, because it's heterozygote, it's showing 10-11, 10-12 in that order. Okay, but here we have what we call the amylogenin, and this what determines the sex of the individual. So here too, it shows that it's what homozygote, meaning it's what XX, and SX is for what female. Okay, so in case it's a male, another peak will show, which will be what XY. Okay, so in that sense, so this is how the DNA profile of an individual. So after, let's say, this is a daughter. Okay, you want to check the paternity of this one, a daughter here. So after you do this, you do same for the dad, and you compare which the alleles the child shares with the dad. Okay, to conclude whether they share genetic markers or not. I hope I'm understood here.
Yeah, all good.
Okay, any question? Okay, so no question. I think it's more technical, but let's try to understand here. So here, the STR loci DNA markers. Okay, STR is standing for what we call the short tandem repeat. Okay, so when you look at our DNA, or when you look at the chromosome, which is made up of our DNA, we have some portion called the loci or the locus. So at this particular locus, normally there are repeat nucleotides. Okay, so that's what is named with the DNA markers. So these are chromosomes. You know, we have 23 pairs of chromosomes. Okay, so let's say with the DNA marker T-pos, this shows that it's on chromosome 2, and D3S1 is on chromosome 3. Okay, now more DNA markers have come about, but initially the FBI, they were using the 13 codex core STI loci. Okay, so this 13, but more have come to bring about discrimination. So here, this is what the short and then repeat means. So with a particular loci where the DNA marker is, so let's say the nucleotides that are repeating, or in other words, the STR, in this sense, example is 8ATG. So this is repeating for 7 times. So this is trying to show that, let's say for this session, it is AATG, this session, the same. So the number of repeats is what is counted with the help of the fluorescent dye that is attached to it. So it will show the number of repeats. And for each individual, I keep on saying you have two alleles because you inherit one from your mother and one from your dad. So it will show the repeat of your two chromosome, or in other words, your two alleles. Okay. So an example of the marker, the nomenclature of it. Okay. So the D stands for DNA, 16 stands for chromosome 16, S stands for single copy sequence, and 539 is on the 539 locus described on the chromosome 16. Okay, so it means that this D16S539 is on what? Chromosome 16, as you can see here. All right, so this what when we talk about STL loci, it means short tandem repeat loci DNA what markers. I hope you are cool. Hello.
Yeah, we are following.
Yeah, we are following.
Okay, sure. So the next one, okay, someone may ask, how does a DNA report look like? Okay, so this is how a DNA report looks like. The red portion here is to show to help in my explanation. Okay, but this is how typically a DNA report looks like. So these are the locals, the one I was explaining, or the DNA markers. Okay, so this an alleged father, mostly the name is here, the child's name is here, but due to confidentiality, I can't pick anyone's reports to show you. So I picked this from internet just to give an idea of how a DNA report looks like. Confidentiality is very important when you're a DNA analyst or anything of that sort. So this is how it looks like. So this child, everyone has two alleles. I keep on saying this child that will take two alleles because one is inherited from the mom, one is inherited from the dad. The father to the same, inherited from the mom, the dad. So two each. But what it does is it compares which allele sizes they share, or in other words, the STR they share. Okay, to prove that indeed an alleged father is the biological father of a child or not. So if the alleged father is the father of the child, you see that exactly half okay, of the allele size or DNA is matching with that of the child. So let's compare here. Here, the dad is having 16, 17, okay, and the child is having 16, 17. So if you are to add the mother, you find out that the child picked maybe let's say 16 from the mom and maybe 17 from the dad. But because we are not comparing with the mom, okay, that's why it looks here that the child is having 16, 17, here to 16, 17. Okay, so here 16, 17, it could mean that the child picks either 16 or 17 from the alleged father as is showed here. And the next one, 16, 17 to the same for the DNA marker VWA. And the next one, 9. 9 here means it's homozygous. So here is what? 9, 9. Okay, but because it's homozygous, we just keep 1. So 9, 9. So here too is showing that the child picked 9 from the dad. And here 10, 11 is showing that the child was picked 11 from the dad. So here the dad's own is 11, 12. So it picked the 11 rather, okay. And when it comes here to the child picked what? 11 from the dad, and here to 8 from the dad, here 29 from the dad, here 11 from the dad. Okay, so when you should do all to this, you find out that um, the child's exactly half of the child's allele is matching with the dad. Okay, and the amylogenin is what shows the sex of the individual. So here S shows that here is a daughter and not son. Okay, so here is SY, father. Definitely, father will have SY. Okay, so this is what it shows. And we have something we call the paternity index. That's the PI. So this shows the so this for statistics. Okay, it shows the how possible is it for a dad to be considered like the biological father of a child at a particular locus. So that's for the paternity index. Okay, so with the paternity index, the product of all these is what we call the combined paternity index. Okay, and the probability of paternity, as I said earlier on, is mostly 99.9 something or 0%. So with this, it literally means that you are the father. Okay, so this is how we report it. We don't report it that, oh, you are the father or you are not the father. Okay, if it's a positive test, this is the way you go about it. So you say the alleged father is not excluded as the biological father of the tested child. So you use not excluded, meaning the father is included, okay, is the biological father. And this is based on the reason that it's based on statistics. Okay, so based on testing results obtained from analysis of the DNA loci listed, the probability of paternity is 99.99. So, okay. And this probability of paternity is calculated by comparing to an untested, unrelated random individual of the Caucasian population. So if it's Africa, since I just picked from the internet, so for ours, we'll use Africa. Okay, if the person is in Asia country, maybe Asian country, whatever it is, all right. So assuming the probability, the prior probability equals 0.5. So this is for statistics reason. So literally, this shows this is how a positive test will look like. So it means that the alleged father is indeed the biological father of the child. Okay. So are you listening? Do you get it?
Yes, thank you.
Okay, sure. So with a negative test too, yeah, this is quite technical. So I'm trying my possible best to let you understand the job we do. In other words, yeah. So with the negative test too, okay, so these are the locus, and you could find we are all sons and daughters of Eve and Adam. So definitely there will be some allele sizing matching. But the moment the child and alleged father doesn't have any relationship, you will see some of the markers, okay, showing nothing to match. So example here, the child is having 11, 12. The alleged father is having what? 8, 9. Okay. So there's nothing showing nothing showing where the child inherited from the dad. So this is giving a clue that there could be a probability this is not the father. So you move further with all the allele sizes, and you could see that there are more to go. Here to 8, 9. Here's what 6, 9.3. Here's 11, 17. Here's what 15, 16. So there's nothing showing that they share genetic markers. So you do for same, but let's come. This is the mother. Okay, mother. So the mother has exactly half of the 8 allele sizes matching with that of the child. Okay, so here you see 14, 15, there's 14 here, 18. So here is sharing 18, 18, here 13, 13, and here what? 10, 10, 12, 12, 15, 15, 30. So in that order, you get it. So it shows that the mother is indeed the mother of the child, but the dad is not. So literally, you are not the father. So here, the combined paternity index will be what? 0. And the probability of paternity to be what? 0%. So in this sense too, with the negative test, you just don't go like, you're not the dad. This is how mostly we report it. The alleged father is excluded as the biological father of the tested child. This conclusion is based on the non-matching alleles observed at the loci listed above with a pi equals to what? 0. The alleged father lacks the genetic markers that must be contributed to the child by the biological father. Okay, so the probability of paternity is then what? 0%. Okay, so I hope it's understood.
Yeah.
Yes. Um, So with this, supposing let's say from crime scene, okay, we have the profile of a mixture from an evidence sample. So maybe you go to a crime scene, you pick blood sample. So what happens at crime scene is normally there are mixtures of samples. Okay, so let's say a murder scene, definitely the victim's blood might mix with that of the perpetrator. So let's say this is a mixture from the evidence sample, and this the suspect standard sample, the profile for it. So you then compare. So comparing, you could see from the top the mixture from the evidence sample that it contains mixtures of profiles. But here, so here it means that the suspect DNA comparing here there's what 15, 15. Yes, 15, 15, meaning it's homozygote. So 15, 15, 15, 15 here, and here to what? 9, 9, 9, 9. You get it? The same. So this could show that there's a probability that the suspect, was somehow had something to do with the crime, was involved, not necessarily that he did it, but there's something involved there. So further investigation can be done to either convict or that acquaint the person of such situation. So now, with the machine that is used for Gene Lab Molecular Diagnostic Limited, this is the machine we use here at Gene Lab. So, what's the 27? We use 27 STR loci marker. So as I said initially, the FBI they were using 13, okay, and some labs are using like 16, some 23, but this is more advanced. Okay, it's using 27 STR loci markers. So these are the markers this machine helps to discriminate. So the larger the number of loci used, the more powerful the ability to discriminate between individuals. So this is what we use at GenLab. So in other words, it gives accurate results. Okay, so anytime you have any paternity case, you bring it to or any kind of relationship test, you bring it to GenLab. We are sure to give you accurate results. So here, this is an idea of it, and this machine is the first to be used in West Africa here as we are using it. So it really gives accurate results. Okay. So the larger the number of loci used, that shows that the more the powerful and ability to discriminate between individuals. Okay, so to proceed now here, quality assurance, this is applied everywhere when it comes to lab work. So this simply means that the process of reviewing analyst reports. So the report or the results you have to check for any maybe in case there's any error. They're saying that two hairs are better than one. You get it. So you look for other people to look through, like with your colleagues, the analysts, to go through, okay, for any to make sure that everything is accurate. So if this is to be performed with another lab, they should also find the same result. It shouldn't deviate from yours. So this is what it's about, to check for technical what accuracy. So that's the last step here. Yeah, so I hope I'm clear with the steps. We are done with the steps. Hello, hello.
We are getting you.
Yeah, so I'm done with the step now. So now let's come to evaluation and conclusion. Yes, I got to realize that when it comes to the technical aspect, most people were quiet. But anyways, you asked for it. So yeah, so evaluation and conclusion. Now I just want to ask a general question. Do you think informed consent is needed when taking samples for paternity tests? Give your reasons for your answer. So can anyone tell me whether informed consent is needed or not? Yes, anyone?
Yes, I think it is needed because it brings
Why?
about trust.
It's what? Come again, please.
Brings about trust. If the person is unaware that you took the sample, the person might not trust your results.
Sure. And moreover, it's to even protect you, the analyst, the DNA analyst yourself. Because sometimes, do you know sometimes some men, let's say a man comes, uh, not always that a man wants to let the result come out positive. That's the child and him, they are sharing genetic markers. I've got this, but sometimes people are getting divorced. Okay, so they are praying that the test will come out negative to prove that maybe their ex-wives were unfaithful. That's why they want to actually divorce completely or something of that sort. So everybody and the reason they come for paternity test. Okay, so you make sure you take informed consent. So sometimes the requirement for DNA, apart from the consent, you even have to take pictures. Okay, you have to take pictures. You have to take any ID, and you make sure that on the ID, the face of the person matches with the face on the ID. And this is to prevent any legal issues in the future. Okay, some may bring someone else. If not that, someone might bring someone else, knowing that this person, when I bring, I'll get a negative result and bring the person. So you do the test with a clear mind, thinking, yeah, then later they find out that they'll be like, yes, I came to do it and it was negative. Whereby maybe in the actual sense, if the father was to bring the biological child, it could be what? Positive. So it's very necessary to take informed consent. You let the client sign everywhere as much as possible, even on the photos, everything. Okay. Yes. So I hope I'm understood at that point.
Yes.
Sure. And what are the best ways to avoid contamination when taking samples for DNA? Can anyone tell me? What are some of the ways to avoid contamination when taking samples for DNA?
By wearing gloves.
And what else?
Maybe probably wearing nose mask.
Yeah, wearing nose mask and maybe a hair cap or to prevent any contamination as possible and avoid talking too much when taking the samples. So you just have to be careful that the samples, the DNA samples are not mixed in any way or one way or the other. In conclusion, DNA analysis plays an important role in various fields apart from forensic. So not only does in forensics is DNA analysis very important in various fields because DNA forms the core, central portion of our being or an organism. Okay, so it forms a core basis in other fields too, such as medicine. Okay, it helps. I think it will get to a time what we call something precision medicine, whereby people will be given drugs according to their genetic makeup. Because sometimes if they make it general like that, maybe a drug that will work for you will not work for me because we have different genetic makeup. Okay, so more research is going into that, trying to find ways that the appropriate medicine will be given to people according to their genotypes. Okay, and also in agriculture, when it comes to genetically modified fruits and all that, it helps. And what we call the genealogy, so for ancestral analysis to maybe know your ancestral lineage and all that, it also helps. And genetics, okay, to know how you inherit certain genes from your parents, how its genetic conditions come about and all that. So this also DNA analysis plays a vital role in that. We have the epidemiology to know the incidence, how to prevent diseases, its causes, and all that. So DNA analysis plays an important role in all these fields too as well. So thank you very much for your keen interest and your listening and all that. I really appreciate. So now it's time for questions. If there's anything you don't really understand, you are free to ask and I will reply.
Yes, please, knowledge. You can ask your question.
Okay, thank you very much. If I'm allowed now.
Sure, sure. Timothy.
All right. So thank you very much, Hannah, for that very lucid talk. Although the technical aspect of it is a bit difficult for me that is green to
Yeah, sure. But
analysis.
some have asked for it, So I think in a way to help everyone get what they requested. Yeah. So sorry for that.
Well, thank you for that question. Actually, my interest is because I'm in medicine and I'm interested in genetics. So I just want to learn about the analysis of DNA interpretation and so on. So that was what drew my attention here, and I'm really very happy I joined and I feel I gained substantially from DNA structure and everything. So please, and you make your email address available on this slide, and maybe we can ask further questions, and you can
Okay,
give.
sure. I'm
Thank you.
available. All right, so I'll make my email available to you and my number in case you have any question, anything you want clarification on. I'll be really free to help. Okay.
I'll be sharing her email in the chat box. She's also on LinkedIn with the same name, so you can check and follow her on LinkedIn.
Thank you very much for the presentation. But I have a lot of questions here. The first question I can ask here is why DNA said the golden standard in forensic investigation?
Please, can you speak louder a bit? I couldn't hear
Okay, I
you.
say thank you very much for your presentation.
My pleasure.
Yes. So I have a lot of questions. So I'll ask one by one.
Okay,
The
okay.
question is why they say DNA is a golden standard for forensic investigation.
Okay, so it's a standard for it because, as we said, DNA is unique. Each individual has a unique set of DNA. So my DNA is different from yours, okay, and it doesn't change. So your DNA from when you were young, when you grow up, it still remains the same. So it's good for to use DNA as a standard for analysis and investigation purposes. I hope I've answered your question.
Not good, but golden.
Okay, so what do you want to know with that?
Why exactly they say DNA analysis is a golden standard? It's like there's double standard in it. How? Where is double standard coming from?
Or do you have anything to contribute to that?
Yeah,
Since
hello.
maybe you want some, yeah, hello.
Yes. I just want to give a little contribution. Okay.
Okay, sure.
Right. He wants to know why DNA is regarded as the golden standard, right?
Okay, sure.
So the correct phrase is the golden standard for human identification. Okay.
Okay.
And it is the golden standard for human identification purposely because majority of the methods used in DNA identifications are validated methods. They are internationally standardized and validated method. It is not an optimized technique or adapted technique for identification. So all the standards or all the standards and the protocols that are followed in the DNA processes, just like you demonstrated, are well validated protocols. Okay. For that
Oh,
purpose,
okay.
when it
Yeah.
comes to human identification, specifically just human identification, DNA, the DNA process remains the golden standard for identification. Earlier, I gave a little contribution regarding fingerprint. Even though fingerprint is also unique, just like DNA, majority of the techniques employed are not well validated, especially because of the subjectivity in the process when it comes to fingerprint. But then the same cannot be safe, mentioned for DNA, which is why when
Oh,
it
okay.
comes to human identification, it is regarded as the gold standard for identification.
All right. So thank you, Mr. Abedi. I hope he
Yeah,
has answered your
that's
question then.
okay.
It's clear now.
Yeah, he answered partly.
Or if
Where
you have
about the
any
double standard? Where it's
contribution,
coming
because it
from?
seems you have you are expecting a particular answer. So maybe if you have any contribution, you can cut across so that we also learn.
Yeah, it's like when he answered yes when he said golden standard because of the labs that to be validated, the accreditation. I think that it can be a correct answer. But where about the double standard? Where double standard coming from?
Is that a follow-up question?
Hello?
Yes, we
Yes.
can hear
Is
you.
it a follow-up
Yes,
question?
I'm hearing a noise. Maybe yours.
I'm asking if
Hello?
you're asking a follow-up question.
Yeah, I'm saying you have answered yes. That's correct. On the, I think it's correct on the issue of golden standard, but on the issue of double standard, I think there's still an outstanding. It's an outstanding issue.
I'm struggling to get what you mean. The issue of double standard has done what?
Golden standard, yes.
Yes, I explained that of the golden. The term golden is just a terminology. So the what it means basically is because the techniques or the protocols that are used in DNA identification, right from the collection to the examination, are all well validated and standardized. Some of the laboratories would have to have accreditations, ISO accreditations, and etc. All right, and these are the protocols that make the techniques applied in DNA examination a standardized protocol. And then for that purpose, we term it golden because now the procedure that GenLab is using is not different from the procedure that a DNA lab in America would be using. They are using the same standardized and validated protocol. Okay, so it is the golden standard for human identification. Now you are asking about double standard. I don't know, and I'm asking if it is a follow up question. How do you mean by double standard?
It's like when we are learning DNA analysis, we had those terms, the double standard and the golden standard. So I think it was my opportunity to ask about that.
Okay. Now the honest answer is that I haven't heard of double standard yet. I know of golden standard, but then if I am to guess around, it could be a synonym or just terminology that is being used. If the phrase is being used with regards to DNA identification and why it is superior, then regardless of the prefix they give to the standardization, whether double standard or golden standard, then it means that like I said, the techniques that the DNA people use in the examination, right from the collection to the analysis, is well validated. That is the most important term. The techniques are validated. All right. So it is well validated and accepted, and they have given a common protocol to be used for the examination and interpretation of DNA result. So if you are to establish a DNA lab right now, you cannot adapt a different protocol for your examination just because you have established a different lab. You would have to go by the standard protocols if you want your result to be admissible in the court law. Okay. So the double here, you need see for the process to be validated, it has to be tested by different scientists. Okay. So it has to be tested by different scientists in different countries, etc. If it becomes clear that the results are reliable and then reproducible, then the particular technique can be validated for use in forensic identification. All right. So the double standard here could be because the protocol has been tested by different scientists producing similar or same result for that purpose, accepted for uh, use in operational laboratories. Okay.
Okay.
Okay, so just
Thank
for
you.
my
I think my
Are
question
you
is
okay now? Are you okay now? Okay. So thank you, Mr. Abedi, for the clarification. All right.
okay, I think
So
we have
any other
someone
question?
we have someone who wants to ask a question. Please, Munir,
Okay.
please, can you ask a
Yeah.
question?
Munta, Hannah.
Yes.
Now I'm checking something. Could he mean double stranded or standard?
Okay.
I'll give you this opportunity. Is it double stranded DNA or double standard? Because there's nothing like double
pregnancy.
standard. I know of golden standard. So you could check and clarify that and then explain more if it is double stranded.
double standard, not double stranded.
Stranded.
Oh,
Double
okay,
stranded
okay,
DNA is
okay.
double stranded, yes, but the issue of yes, standard,
All right,
I
all
think,
right.
is like when you are doing DNA analysis, where the weight coming from is like you have two standards. Like we have allelic ladder and the least 600.
Okay.
You get it?
Yes. So see, so sometimes you have to be, if you are talking about standard in terms of what we would call it a reference sample or a known sample, then I think you have to be clear. But then if you ask double standard following a golden standard, then it makes the understanding quite challenging. Okay. So if it is standard, as in the standard they would use or as reference for the examination, then probably he could add more light to it. But if it is standard with regards to the golden standard that I explained, then I can't give an explanation for double standard, right?
No, the issue is
So
we have two
I don't know if
terms.
they use a standard for the examination. Maybe Hannah can explain more to that.
Hello?
Yes. Hello.
Because of
Yes,
time
it's
factor,
like we have two.
maybe we can allow Mr. Knowledge. Maybe if he goes back and then he gets things well organized, take a follow up, then we discuss it.
Okay, so the others can also ask their
So let's
questions.
take a few questions, then we can close. Please, Mustafa Munir, if you are there, you can ask your question.
Yeah, thank you very much for giving me this opportunity. Please, may I know at what stage of pregnancy can DNA be carried out for paternity confirmation? That's the first one. And then the second one, is there any technology that we can use fingerprints to trace the DNA of an individual? Thank you. Is there any correlation between fingerprint and then the DNA makeup of an individual so that you can use that one to trace the person's background? Thank you.
Okay, thank you. With the pregnancy, okay, I think for that with within as early as 16 weeks, you can use the paternity with the amniocentesis. Okay, so the stage, in other words, the stage in pregnancy whereby the amniotic fluid will start producing. So with that, you can use the amniotic fluid. You can get from that and use for your paternity testing. With your second question, whether there is any correlation between fingerprint and DNA, whether you can use that to find any correlation. Am I right?
Yes, madam, you're right. Whether I can use someone's fingerprint to trace the parents.
Fingerprint? No, no, no. I don't know of that. Honestly, I've not heard of that. Whether we can use fingerprints to trace with that, no. But what I know is okay. You know, we have types of fingerprints. We have the three main types. We have the arc, the loop, and also the hole. Okay. What I know is we can inherit the type of fingerprints. But when it comes to the actual ridges or the miniature of the fingerprints, we don't inherit them from our parents. It is due to the environmental condition in the womb that we we get the particular ridges or, lines or whatever we have on our fingers, meaning the fingerprints. Okay, so it's kind of mutually exclusive. They all come differently, but in investigation, they can help each other. Okay, to link a particular suspect to a crime or whatever it is. I hope I'm making sense here.
Okay, so adding up to that,
Okay.
if I'm not getting rusty, the one we ascribe to as the father of fingerprint analysis, the one who brought about the using the finger minutia to distinguish between people, that is Francis Galton. During his early days of his work, before he came to that conclusion, his aim was to get some kind of genetic relationship between fingerprints. But at some point, he realized that there was no any form of correlation. So he dropped that, even though he discovered that the fingerprint was the unique identifying feature, which is kind of very distinct and unique for each and every person. So there is no until now there is no any studies that shows that there is a correlation between the fingerprint and the genetic makeup.
Okay, sure. So more research will go into that, I guess. Maybe Muniru, if you're into that, you can research into that. That will not be bad. But as for now, as I know, there's no correlation. That's what I will say. I hope you're okay.
Okay,
Okay,
madam,
so
thank
Dennis, if
you.
you are still around, you can ask your question. I think Dennis is off.
Okay.
Someone was about to ask a question. Okay, so if there's no other question, I think we can end today's session.
Okay. So, okay. Then the closing remarks. Can we have?
Yeah, okay. Before we close, I would like to apologize for the inconvenience caused I think we will take constant measures from here on to ensure that it doesn't happen in future lessons.
Okay. So
Okay.
thank you all for
So
your
thank
time
you
and
all for your
attention.
time and attention.
We appreciate your
We appreciate your
participation,
participation,
and then we hope
and
that
then we hope that
from time to time
from
we can
time to
come
time we
together
can come together
to
like this
share
to learn and
ideas
share
on the
ideas
areas of
on the
forensic.
areas of forensic.
A very big
A
thank you
very
to
big
our
thank you to
speaker.
our speaker,
I think you did
and
a
I
very
think you
good
did a
job,
very
even
good
though with
job,
the
even though with the
challenges.
challenges.
I've learned a lot
I've
from
learned a
your
lot from
lessons,
your lessons, and
kudos to
then
you.
kudos to you.
Okay, hoping to
Okay,
see
hoping
more
to
from
see
you.
more from you in future.
Okay,
Okay,
so I would
so
like to
I would
add up.
like to add up.
The session is being
The session is
recorded.
being recorded. If
If we finish, I'll
we
try to
finish, I'll
compile
try to
everything,
compile everything
share
and
links
then share
to
links
those
to
who registered
those who
via the link.
registered via the link.
You can also
You
contact
can
us
also
through our
contact us through
email
our email
or contact
or
our speaker
contact our
for
speaker
the
for
recording
the
and also
recording
the
and
slides.
also the slides if in case of anything.
Okay, so the forensic.
So
Please,
the forensic.
before
Yeah.
the next session coming on, please.
Hello.
Hello.
Before you leave, when is the next session coming on, please?
Okay, we'll
Okay,
update you
we'll
very
update
soon.
you very soon.
We'll update you very
We'll
soon. We're
update you
still
very
in
soon.
touch
We're
with the
still in touch
speaker.
with the
If
speaker.
he confirm the
If he
date, we'll
confirm
let you know.
the date, we'll let you know.
Yeah.
Okay, so
Yeah.
hello,
Okay.
hello,
Yeah. Yes, please
Yeah.
hello.
talk.
Yes, please talk.
Okay, so I was thinking, aside your site, your YouTube site or other site that you guys have to put information there, can there be a WhatsApp group whereby all those who join today can have access to, so that maybe information can be put over there for them to have access to know the time and the date of the next meeting, if possible,
Oh,
so that
great,
you can
great.
know
Oh,
anything.
great, great.
Yeah, we have
Yeah, we
various
have various
platforms. I'll
platforms.
just say it
I'll just
here
say it
for those who
here
want to
for
join.
those who want to join. Yes,
Yeah.
please.
If
Including
Telegram
WhatsApp.
or
Including
if
the WhatsApp
WhatsApp or
platform.
yeah,
Yeah.
so it can be possible so that people can join. And you know what people can join. It will help. Okay, all right, thank you.
Okay.
But
So,
I've enjoyed the presentation very well. Go. We pray next time we have a good network so that we can learn more from our speaker for this evening. Thank you very much.
okay. Thank you, all of
Okay.
you.
Thank you, all of you.
So if you check on the
So if
chat
you check
box,
on
you
the
see
chat box,
various
you see
links
various links
to various
to
pages,
various
including the
pages,
WhatsApp.
including the WhatsApp.
And I hope in
And I
future we
hope
can
in
have
future
some
we
of
can have
our
some of our
audience
audience
being the speakers to
being the
give us
speakers to
lessons
give
from
us
their
lessons from their
how like on
like
the
on various
various specializations.
specializations.
Okay.
Okay,
So
okay.
thank you all
So thank you all
and have a good day.
and have a good day.
Thank you too. Have a good day too. Bye.
Okay, bye.
Okay, bye.
Thank you. Bye bye.
Bye.
Bye.